Hello everyone, welcome to Dr. V.S. Krishna Government Degree College Autonomous Visakhapatnam Media Center, and I am Dr. A.H.D. Pushpa Latha, lecturer in Botany from Dr. V.S. Krishna Government Degree College Autonomous Visakhapatnam. In this session, I am going to discuss about bacterial conjugation. Let us see what is bacterial conjugation. Yes, this is nothing but transfer of genetic material between two bacterial strains which are in direct contact with each other. It is not considered as a true sexual reproduction because after the process it does not generate any new offspring, and rather it is considered as a parasexual reproduction because the existing organism's genetic material gets transformation, and the mode of genetic material transfer is known as horizontal gene transfer. That means there is no parent offspring relation between the participants. Let us see. Yes, this picture shows about vertical and horizontal conjugations or horizontal gene transfers. In the first figure, we can see clearly that the descendants are getting the genetic material from their parental cells, whereas in the down picture, we can see clearly that it is an unrelated organism or unrelated bacterial strain. To this bacterial strain, the pink color strain is transferring its genetic material to the blue one, which are not in parent offspring relationship. If we want to define conjugation clearly, it is the genetic transfer between bacterial cells, that is from one cell to another, which are in direct contact with each other by formation of a cytoplasmic bridge known as conjugation tube, and the bacterial strains or cells which are in connection or in this process are known as conjugants, and one of the participants, that is one of the conjugants, is known as donor strain, which acts as F plus, and the other one is F minus, which acts as a recipient strain, and there is a physical direct cell to cell contact between the conjugants. Yes, in this picture we can see a donor cell or an F cell. It has plasmid or F factor, that is in the cytoplasm. That is, F factor is nothing but it is an extra chromosomal DNA present in cytoplasm. Yes, this red ring is plasmid or F factor. Double stranded DNA and it can replicate its own type. There are three different types of F factors or donor bacterial cells: F plus, F prime or F dash, and HFR bacterial donors. F plus type of bacterial donors are a kind of strains that have F factor or fertility factor in their cytoplasm. In F prime cells, the plasmid is derived from F plus strains and has some genetic or some chromosomal genes present along with F plasmid. In HFR strains, the bacterial strains exhibit high frequency of recombination for certain chromosomal genes. Let us see F plasmid in detail. The F plasmid is an episome, that means an extra chromosomal DNA which is double stranded in nature, which is circular in nature and self replicating structure. It can integrate of its own in the bacterial chromosome by homologous recombination, and its length may be about 100 kilobases, and it carries its own origin of replication, that is ORI-V, and also an origin of transfer, that is ORI-T. And probably only one cell may be present in one copy is present in one cell and carries about 50 genes. Sometimes they may carry more than 50 genes in each plasmid. This is a typical plasmid which has several genes, that ORI-T location is here and ORI-V, which is an origin of replication site or gene. After process of conjugation, F minus becomes F plus, and this is called a complete conjugation or legitimate conjugation. Sometimes conjugation may be incomplete or illegitimate. That means if both the strains that are in conjugation might be F minus, in that case the process is not complete and it results in a non-productive conjugation, and the two cells separate without exchange of any genetic material. That is called illegitimate or incomplete genetic transfer or incomplete conjugation. The process was discovered by, conjugation was discovered by Joshua Lederberg and Edward Totem in the year 1946 in Escherichia coli bacteria. If we start the mechanism, it starts with activation of certain genes, that is initiation of certain genes that are responsible for synthesis of some proteins called membrane binding proteins. These are very essential for binding of F plus cell to F minus cell, very essential, and these are present especially in F plus, that is donor bacterial cells. They activate the and they synthesize the membrane fusion proteins. They enable the attachment of F plus cell with F minus cell. After initiation of such genes and synthesis of the membrane protein, the gene that is present in the cell, that is relaxase. Relaxase is an enzyme that is produced by or synthesized by a gene which creates nick in double stranded DNA. After nicking the DNA, the broken DNA, the nicked DNA separates from the unbroken one and it tends to move from donor to recipient cell in a direction known as 5 prime to 3 prime. Always we know that always the direction of replication is 5 prime to 3 prime, we know that, as well as here also the broken genetic material or the plasmid, the strand moves in 5 prime to 3 prime direction from donor cell to recipient cell, and the remaining strand replicates via rolling circle model. In the picture you can see clearly the rolling circle model replication of DNA, in that one of the strands replicates, one of the strands nicked by the enzyme. After nicking that separates from the double strand, other unbroken strand, and then starts replication. These are the various steps that are in conjugation process. The first step telling us there are two conjugates, two strains of bacteria participating in the process. One of the bacterial strains that has the F plasmid or the plasmid or F factor or fertility factor in the cytoplasm that acts as a donor one, having an extra or additional structures on the surface. They are pili called sex pili. The presence of sex pili is very important because attachment is only done by these sex pili only. These are present in donor cells. There is a difference between donor and the recipient conjugates. In donor conjugates, F pili are present or sex pili are present. They are absent in recipient strains. In this picture, in the second one, after initiation of the genes, after activation of the initial genes, they synthesize membrane proteins, membrane binding proteins, and they enable the attachment of this donor cell to the F minus or the recipient cell. After that, there is an establishment of a bridge between the conjugates, that is donor and recipient. It is the cytoplasmic bridge called conjugation tube. It is called conjugation tube. It is the conjugation tube present between two conjugates, two cells, two donor and recipient cell, established by sex pili because of activation of some genes that are responsible for synthesis of membrane binding proteins. And in the next step, a relaxosome. Relaxosome is nothing but DNA binding proteins. They cause the movement of the nicked DNA from donor to recipient cell. And the next one you can see is transferosome. Transferosome facilitates the nicked DNA from donor bacterial cell to the recipient one. After moving from donor bacterial cell to the recipient one, both the partners, both the conjugates will become F plus after completion of the process. In the last step, you can see clearly both the donor F plus as well as the recipient become F plus donor. That means the donor bacterial cell replicates the one which has transferred to the recipient one, and the recipient bacterial cell starts replicating or synthesizing the complementary strands. In the same way, both the partners or both the conjugates will get the same plasmid. That is why F plus will never become F minus, and F minus transforms into F plus. This is the advantage of this process. F plasmid, it integrated in the chromosome. It integrates in chromosome sometimes. This kind of F plasmids or strains are known as HFR strains, high frequency recombination strains. In these cells also behave as F plus strains equal to F plus, and they form sex pili and undergo conjugation with F minus cells, and HFR strains replicate as F plus strains, as we have seen in the previous slides, that they undergo replication process as like a rolling circle model. Conjugation in HFR strain begins with F plus, that is nicked at origin of replication, that is OREV, and then F plus and bacterial chromosomal DNA is transferred into F minus cell, and just as it does in case of conjugation between F plus and F minus cell using rolling circle model. Yes, this is formation of HFR strain. In the first case, we can clearly see that there is an extra chromosomal DNA in the cytoplasm, and in this the cytoplasm DNA, that is plasmid or F factor, that because of recombination, homologous recombination, it gets integrated into the bacterial main chromosome to form HFR strain. This is how the conjugation between HFR and F minus takes place. This is the actual DNA, chromosomal DNA, and the piece that is represented in red color is F plasmid or F factor, that is integrated in the chromosomal DNA of bacteria, and F minus, it does not possess any plasmid or F factor. After establishment of bridge between these two conjugates, the F factor, somewhere after nicking it, it moves into some of the factor, some of the amount of DNA moves into F minus cell. Here it is establishment of HFR and F minus conjugation tube, and then passing of one of the strands into the recipient cell. After that, after completion of the process, both the conjugates will separate from each other. Now, HFR will going to donate its F plasmid, some of the F plasmid, but it never receives the entire F plasmid in case of HFR strains to F minus. This is another cell, another picture showing different steps of bacterial conjugation between HFR and F minus cells. Yes, that is how much of bacterial chromosome, that is how much amount of bacterial chromosome is transferred from HFR to F minus depends on length of the time, that is duration of time that the two cells remain in contact. And in mating of HFR and F minus, the F minus cell almost never become F plus or HFR, because the factor is nicked here, is in the middle, not at the end. That is why it has to, if it has to enter, if it has to get the whole F factor, then the entire bacterial chromosome must move into the recipient one. This never happens in the nature. To become F factor, as I said, the HFR strain has to donate its entire DNA to the F minus cell, and this event occur rarely because most of the conjugation or the conjugate cells break apart before the entire chromosome has been transferred. When an F factor is excised, that is separated from the bacterial chromosome, a small amount of bacterial chromosome may be removed along with F factor, that is called F prime or F dash. It is a different kind of bacterial donor bacterial cell. This is the process of conjugation, and there are various applications, especially it is used in transferring genes from one organism to another. They act as like vectors, they can act as vectors or transferring agents between cells. That is the first application, and conjugation has advantages over other forms of genetic transfers, and relatively large amounts of DNA can be transferred from one cell to another using conjugation process, and even it can also be involved, it can also be used in genetic engineering process, as we know that there is TI plasmid. Via this plasmid we can transfer genes from one organism to a desired one. This is regarding conjugation in bacteria. I hope you understood well. Thank you.